Okay, let's go ahead and get started. So thank you all for coming. I appreciate it. My name, for those of you who don't know me, my name is Beth Norman. I'm the Director of Science and Research at Lacawac Sanctuary, and I also run the PLEON program. So this virtual workshop is going to share with you all some of the results that we found in the Wallenpaupack Citizen Science Monitoring Program in 2020. So I'm looking at the names here showing up on my screen. I think a lot of you participated in this program. If you did not, we will kind of go through a little bit about what it is and how you might want to become involved in the future. But before we get too deep into the actual data, let's just go over some Zoom guidelines. I think by now all of us are probably getting pretty familiar with this particular platform, but for those of us who may not be, if you don't mind, just try to keep your volume muted unless you're saying something to the group. That way we cut down on the background noises. Your mute button should be in the lower left-hand corner of your screen. I want this to be very interactive. I'm going to try to keep my own talking to a minimum. So I'm really interested to hear what you all have to say about the program and to take your questions and discuss the data. So you can do that either verbally, feel free to unmute and interject or ask a question. You can also use the chat feature if you feel more comfortable doing that, asking a question as we go along. Feel free to go ahead and use that chat. I do have an intern working for me this fall. Claudia France is here. She is going to be monitoring that chat for me. So as you enter those questions, she will make sure that they get to me if I don't happen to see them because I'm looking at my PowerPoint screen or something like that. So she's going to be helping me keep track of your questions through the chat. Also, I believe this should be, yes, this is recording. So my intention is to post this on the PLEON website, this webinar, once we're all said and done, so you're aware of that. So we also have Lauren Nose, who's on this meeting, and she is a PLEON scientist who works with PLEON. She's a graduate student, a PhD student at the university, at Miami University in Oxford, Ohio. She's done a lot of work with PLEON and is on our science advisory board. So some of you may have seen her in previous workshops, but she will be available for questions and discussion and help this move along as well. So, okay, with that, let's get started. My intention is for this workshop to be about an hour. Again, I'm going to go through our some of the data, but I really want the bulk of it to be discussion, question, answer, that kind of thing. I am also happy to stay longer than an hour if some of you have more questions or would like to keep the discussion going, that's fine, but we'll try to get the main points down so that you all can go on with your evening. All right, so with that in mind, I'm going to share my screen here. I do have a couple of slides because I find that data are easier to see when you can actually see them visually. So let me go ahead and share my screen here, which is always a little bit, but there we go. Okay, so you all should be seeing a PowerPoint presentation at this point. Okay. Which may or may not actually work for me here. Awesome. All right, what's going on? Ah, here we go. It wouldn't be a Zoom meeting if something didn't give me a little bit of a hiccup here. Okay. So first of all, let's start with the basics. Like I said, many of you have actually participated in this program, but for those of you who may be new, basically, why community science? Why do this? Why would I put this program together? Why would you participate in this program? Well, first of all, and most importantly, in my opinion, community science programs like this one really connect scientists who study lakes with the communities who live on and enjoy lakes. So scientists have a skill set that's very important. We are trained to look at data, to collect data, to interpret those data. But the communities and people who live on these lakes, you guys are what I call the first responders. You see what's happening in your lake. You're very connected to the changes that are happening. You see when something looks off or something's different. And so while someone like me has the training to maybe interpret some of those changes, it's people like you who really see them, and most importantly, who live with their implications, right? You are on the lakes. This is part of your lifestyle in your lives. And so to be able to include lake communities in the collection of scientific data, I think is really powerful from both perspectives. This partnership and partnerships like the ones that we're building really advance effective lake management and preservation. I mean, that's the ultimate goal. We want to make sure that Lake Wallenpaupack and other Pocono lakes are preserved and managed appropriately. Programs like this can generate scientific knowledge and breakthroughs. You are collecting scientific data, and that is something that we can use to make policy decisions and build on for other scientific purposes. Hopefully, we're increasing lake literacy and scientific engagement within our communities. And really importantly, we're building, I hope, trust between scientists and the communities that we serve. So scientists are part of communities. We're not something separate. We're not someone different. We are part of the community. And I hope programs like this really strengthen that partnership. So we kind of have a pyramid triangle over here, basically how this program works. Those of you who participated in the program took a training in the beginning of the summer to learn how to collect these data. Then you collected those data using hands-on water quality monitoring. And now tonight, this is the fulfillment, bringing those two things together. We're discussing the results and implications. So you understand what data you collected, why, and what we can learn from them. Big thank you to everybody who participated in this year's program. We had a really big growth from 2019, so thank you to all of you. Also, thank you to our funding sources, past and present. So PLEON was started with seed money from the Pennsylvania Department of Environmental Protection. We do get some support, or have gotten some support, from the National Science Foundation. And of course, Lacawac is our hosting institution. All right, so what I'm gonna for how I'm gonna frame this workshop, I wanna go over some data. So hopefully that will be about 15 minutes of me talking. And feel free to interrupt me with any questions you have or discussion points. This is very informal, so interrupt anytime. And then I really want to discuss those data and have question and answering time for you to really ask whatever you've been thinking about the lake over this past summer. I would also like to elicit some of your feedback on the 2020 program, particularly the Lake Observer app, which is the first time, this is the first year we've used this particular app, and I'm interested to see how you thought it worked and if you think it is worth moving forward with it. And I'm always up for ideas for what we can do to improve the program, grow the program for 2021. Okay, so again, feel free to interrupt me, but let's just jump right in here. So here's a map of Wallenpaupack, of course, and these are the sites that were sampled in 2020, and those sites are shown as red dots. We had 20 total sites, which is amazing, up from 13 in 2019, so that's great. 10 sites were in what I'm calling the southwest end of the lake, down here, if you can see my cursor. 3 sites were in the northeast end of the lake. And then 7 sites were in what I'm calling mid-lake. And if any of you are curious on how I decided the sections, pretty much arbitrarily. There's an island right here, there's an island right here, and I put everything between those 2 islands, as I called the middle of the lake, and then the 2 ends. So the major finding from 2020, we had a lake-wide algae bloom, which I'm sure those of you who are on the lake and participated in this program remember quite clearly. So here are some photographs from the summer at Wallenpaupack. And as you can see, and as I'm sure you observed off of your own shores and docks, the lake was quite green. This was the major finding. So let's put some numbers to these pictures, because I'm a scientist and that's what I do, and that's what you have done as well this past summer. So let's look at these data over time. And what I'm showing you is a graph here. On the horizontal axis is time. So we started this program on May 30th. And every other week, those of you who participated in this program will remember, every other week you brought me a water sample. From that water sample, I looked at the algae abundance in the water. And I did this using an assay for chlorophyll A. Chlorophyll A is a compound that is found in algae cells, and it's just the way that we quantify algae in the laboratory. So I can extract chlorophyll A from the water sample that you brought me, and I can quantify that in the lab. And that's what I've done here. So on the y-axis, or the vertical axis here, is just the measure of chlorophyll A concentration in micrograms per liter. All you really need to know about this is the more chlorophyll, the more algae. So this is really algae abundance on this vertical axis. And what is very clear over the course of the summer through June, July, and August, May through July, you know, we had pretty stable algae. And then towards the end of July, boom, we went way up, right, in Wallenpaupack. And these bars are averages from all of the water samples that were brought to me on that particular week. So they're across sites across the whole lake. So we can see basically in number form what you can see in these pictures here. Lake is green because there's lots of algae in it. I also worked with a young man who did this program in another lake in the Poconos. And I'm going to show you those data right here. So in this other Pocono lake, he did the same program as was done on Wallenpaupack. And what we saw is a very similar pattern. You know, fairly stable algae abundance early midsummer, and then an increase in algae towards the late end of the summer in that other lake as well. What you also might notice is that Wallenpaupack had a lot more algae than this other lake. So although the two lakes were following a similar trajectory, the bloom in Wallenpaupack was much more severe. There was a lot more algae, about twice as much algae in Wallenpaupack than this other Pocono lake. Even though we saw similar patterns, Wallenpaupack bloomed with a lot more algae. So that's the first take-home message. The second take-home message is we can break down the sites in Wallenpaupack into those three sections I described earlier: the southwest end of the lake, the northeast end of the lake, and then that middle section of the lake. So instead of showing you all the averages of all the sites, here I've broken it down into those sections. And what you see is that in all of the sections, we saw that similar pattern: low algae in the beginning of the summer, and then in late summer, we saw that increase in algae. But if you look at that 22 August sampling point, this blue bar represents the amount of algae found in the southwest end of the lake. And you see that that's a lot higher, about twice as much more algae than we found in the northeast end and in the mid-lake section. So although we saw the same pattern over time, the bloom at the southwest end was much more severe within Wallenpaupack itself, right? Now there's a couple things to keep in mind when you're looking at both of these graphs. And that is that Wallenpaupack is very different from other Pocono lakes. Wallenpaupack is the third largest lake in Pennsylvania. It's certainly the largest lake in the Poconos. It is very heavily used. It's very heavily developed, and there's lots and lots and lots of recreation on the lake. I'm not telling you anything you don't know. So while this graph is comparing Wallenpaupack to another regional lake, it's a little bit like apples and oranges, because that other Pocono lake is much smaller. It is developed, but not nearly to the extent that Wallenpaupack is, and it doesn't see nearly the amount of traffic that Wallenpaupack does. So it's just something to keep in mind when you're looking at these data. This comparison is interesting, but it's not perfect. Same thing when we look at this other graph. We do see this more dramatic bloom on the southwest end. Something to keep in mind, though, is that that average was probably 7 or 8 different sites, while the bar that is describing the north end was only 2 sites. So we have a little bit of a sampling bias towards one end or the other. So I can't say that this is ecologically significant finding, but it's an observation at this point, is that we see that that tends to be more algae in the southwest end than the northeast end. Any questions so far? Are we all on the same page? Okay, so we saw that algae bloom.
Beth.
Yeah.
Beth. The other unique thing about Wallenpaupack is it shrinks considerably in volume between the first 2 months of the year, June and July and August. I know the temperature is rising, but it shrinks by hundreds of millions of gallons, which the other lakes don't do. And the southwest end of the lake becomes extremely shallow, as all of the boat owners know.
Yeah, absolutely. And Wallenpaupack is heavily managed, while as the other Pocono Lake is not as heavily managed. And that change in volume can absolutely impact algae communities for sure. So that's an excellent point. Again, a little bit like apples and oranges. And it is helpful to look and see that, okay, you are seeing increases in algae over the summer in other lakes. What makes Wallenpaupack different? Why is that bloom more severe? Is it because of the volume change? Is it because of the size? These are all things that we can look at. But yeah, absolutely.
Hi, Beth. It's Tina. I have one question back to the charts. The spike in June, can that be correlated back to a rise in temperature? I think it was warmer there for like a week in June.
Yeah, so that's something that we can look at. We certainly have those data. It's a little bit tricky at this point.
Or rain.
Yeah, right, exactly. So what we want to try to do now, or moving forward at least, is to start asking questions about what environmental factors are contributing to these blooms. And to do that, you really need real robust data. And we're starting to build that data set. You not only want a lot of data taken on the same lake at the same time, the way this program does, but you want to be able to look back across years to see if those relationships hold up. So we're not quite at the point with this program, which is very young. This is only the second year we've been doing this. We're not quite at the point where we can start making those correlations, but that's where we're moving towards, if that makes sense. So that's a good question.
Yeah,
that is what I would want to see.
Up and then it really it dropped off in July.
Dropped back down.
I was just very curious as to, what could be driving that.
Yeah, it could be temperature for sure. It could be precipitation. So that's another thing to look at is what were the rainfall patterns. Sometimes rainfall can affect algae in lots of different ways. It can stir up the lake, which can bring nutrients, which algae need to grow. So sometimes you might see a bloom following a rainstorm, but it can also, dilute the lake. It can do a lot of different things. It can cool the lake down, depending upon what time of year it is. So precipitation, temperature, I would say for Wallenpaupack, certainly the pattern of drawdown would be something we would want to look at. And as we build this data set, I would say within the next 5 years, we'll be able to start really doing some robust analysis to that end.
Thank you.
Yeah, absolutely.
Dr. Norman, someone in the chat just wanted to double check that depth plays an important role in this as well.
Yes, yeah, absolutely. So depending upon where you are in the lake, we might see different patterns. As Bill mentioned earlier, the drawdown of Wallenpaupack really is seen at the southwest end. So that is something that could be potentially affecting algae populations. And again, the more data we're building over the next several years, we'll be able to start putting correlations and really nailing down some of these really great ideas. But we just need a little bit more data in order to make some robust comparisons. So we saw the algae bloom. And you could probably tell by looking at these pictures, there was a corresponding decrease in water clarity. So the algae bloomed, and there's lots of different things in lakes that can cause water clarity to decrease. Algae is one of them. And in the case of Wallenpaupack, we have a pretty strong case for the decrease in clarity that we saw being due to algae. So I'm going to show you graphs of Secchi depth. So for those of you who participated in the program, you remember going out with your Secchi disc, which is that round disc that's got quadrants of alternating black and white. And you lower that disc down into the water until you just barely can't see it anymore. And then you measure that depth, and that's called the Secchi depth. And you can imagine that a really crystal clear lake, you'll be able to lower that disc down quite far before it disappears. Whereas in a lake that's more murky or less clear, you won't be able to lower it as far. So this graph is oriented a little bit differently than the previous graph, and that's just because I think it's intuitive. So if we look at the horizontal axis, again, we have time. So we started on the 30th of May, we went through the 29th of August. Secchi depth readings were taken every week. And the horizontal and the vertical axis is that Secchi depth. So the way I orient these graphs is so you can think of this 0 as being the top of the lake. And you lower that disc down, and the farther down into this cartoon lake you go, the more clear the water is. The higher up, the more shallow that bar is, the less clear the water is. So this is, again, the average Secchi depth of all the sites across the lake over time. And we see a pretty similar pattern to what we saw with algae, whereas we have a certain clarity or certain Secchi depth between 2 and 1.5, pretty much into the middle of July. And then we see a decrease in clarity right about the same time that we saw that increase in algae abundance. So what we see is that it is very difficult to see through this green lake. This green lake is not very clear, and we see that in these data. We can break that down just like we did previously. We can break that down into the lake sections, the northeast section, the southwest section in blue, and the mid-lake section in gray. And again, we see the same pattern in each one of those sections, but we do notice some differences. These yellow bars tend to extend farther down into the lake, meaning that the Secchi depth was deeper and the lake was more clear in the northeast end. That's what the yellow bars are in the northeast end. Whereas the southwest end, these blue bars tend to be more shallow, even in the beginning of the summer. We then see both sets of bars and the mid-lake bars decrease as that algae bloom kicks in. But that decrease is much more substantial on the northeast end. So this is what I mean by that. If you look at sort of the average Secchi depth at the northeast end in the June, early July, and you compare that with August, on average, that changes about 1.25 meters. That Secchi depth decreased 1.25 meters in the northeast end. If you look at that same decrease in Secchi depth at the southwest end, you're only losing about 0.3 meters. So that change is really being driven, at least according to these data, by the change in the northeast end of the lake. But huge caveat here, we had a very small sample size in the northeast section of the lake. So again, those averages are based really on, 2 sites sampled in the northeast end, whereas the southwest end, we had far more sampling sites. So we're looking at 7 to 8 values going into those averages. So again, there's a huge caveat that comes with these data, but it is interesting. So one of the things moving forward as we build this data set and we go year to year to year, is this difference consistent, right? Are there true ecological differences between the northeast end and the southwest end that we're starting to see glimpses of in these data? Okay, so we saw the bloom in the laboratory data where we actually looked at chlorophyll, the algae abundance. We saw a corresponding decrease in water clarity. The third big take-home message from our data is that we saw these algae, you all saw these algal blooms in the visual qualitative data that you took. So one of the things that you were able to assess was the water color. So you looked at the Secchi disc and you used it to determine what color the water was. And we gave you a couple of options. The water could be brown, blue, blue-green, yellow-green, green, or clear, right? And you recorded those data each week. So what this, it's not really a graph, it's more like a color map, is showing you each one of these rows going across is a single site. And I broke them down by the northeast end, the mid-lake end, and the mid-lake, and then the southwest end again. And then across the top, you have the date. So, and then I just color coded each cell based on what your visual observation was. First thing that jumps out at me anyway are these gray cells, and that means that no data were recorded. Okay, so anytime you see a gray cell, that means there's no recorded data. The other thing that you can sort of see from these data, if you squint, is that typically the brown is showing up early and the lake is looking a little bit more green as we move later in the summer. And again, that was really evident just from the chlorophyll data as well as the Secchi depth, is that we saw that shift towards more algae in the lake. And you saw that in your qualitative data. Okay, so we are 2 years old on Wallenpaupack. So we can compare 2020 to 2019. We had more data in 2020 than we did in 2019. But if we make that comparison, what's interesting is we saw blooms both years. So this is, again, looking at the amount of algae in the lake, using chlorophyll A to measure that. We have the summer going across the horizontal axis. 2020 data are in the filled-in circles, and 2019 data are in the open circles. So we see a similar bloom somewhere in the summer. Chlorophyll A went through the roof, right? And for those of you who remember last year, I remember the lake turned green. So we see that in the data as well. What's interesting is that the 2019 bloom started earlier in the summer, but the 2020 bloom seemed to be more severe. There was more algae in 2020 than there was in 2019. So what, moving forward, what is an interesting question is this typical? Is this becoming the new normal or the normal for Wallenpaupack? Are you going to always see these midsummer blooms? And full disclosure, I moved here in 2018. So those of you who've been on the lake for a long time would be able to answer this probably anecdotally. Do you always see an algae bloom? Are the lengths of these algae blooms changing? These are questions that this data set will be able to answer as we keep moving forward. Are they starting earlier? Are they lasting longer? Are they becoming more severe, or are they decreasing in severity? And as we talked about a little bit earlier, we can start to correlate these blooms with environmental factors. So the more data we gather, we can really start looking at the effect of temperature. We can look at the effect of depth or drawdown. We can look at the effect of precipitation and some of these other nutrient concentrations and some of these other environmental variables that we can start making correlations and think about describing what is the perfect set of conditions that prompt a bloom in Wallenpaupack. Then you can start thinking about what can we do about that, right? But we have to understand what's driving these blooms first. Okay, so that was a really fast crash in what we actually found as far as data is concerned. I didn't go over every single variable that you all collected for the sake of time. I am writing up a report for you so that this will all go into the report. You'll be able to look at each one of the variables. The 2019 report is on our website. So if you're interested in what was found last year, you can check that out before I get the 2020 report up.